Hi guys, I've decided to make a video of the chapters just to quickly review the main subjects that you have to remember. So chapter 3 is about the macromolecules. So let's move to the next slide. Generally, in this chapter, the focus is the definition of organic compounds, and then we're going to focus on macromolecules that are divided into carbohydrates, lipids, proteins, and nucleic acids. You need to remember these four types of macromolecules. Now, what is an organic compound? Organic compounds are based on carbon molecule. Carbon is an element on the periodic table, and it has 4 electrons in the outermost shell. So that means it likes to share electrons with other elements and to form covalent bonds. So that's pretty much all what you need to know about the organic compounds. And then we have also hydrocarbons. That means these structures are a combination of the carbon and hydrogen atoms. And that's all about hydrocarbons. There are also larger hydrocarbons. Again, I'll let you read through this. Important is also to know about the hydroxyl group and carboxyl group. These are called functional groups that make a difference in the chemical characteristics of the elements or food items or anything around you. Could be hormones, could be food items. So they all carry or attach to these functional groups, and they give these items a special characteristic. So again, these are examples: hydroxyl, which is OH, and carboxyl is COH. Let's focus on the carbohydrates and lipids, proteins, and nucleic acids. So when I ask you what are the 4 major macromolecules, you immediately have to say carbohydrates, lipids, proteins, and nucleic acids. And you need to know these are macromolecules. So that means they're giant molecules, macro. And obviously anything that is macro is made of micro. So they are made of smaller monomers. Mono means one, and these guys are polymers. Poly means many. So how do we make polymers? Obviously, if we add the monomers, so imagine you have one Lego piece, attach those individual Lego pieces together to form a polymer. However, in order to do that, like from one monomer and another monomer or shorter polymer to make a larger polymer, you have to remove a water molecule. So if you remove the H from a monomer and the OH from another monomer or a polymer, you will end up forming a longer polymer. So this process is called a dehydration reaction or dehydration synthesis. D means removal, hydro means water. So a water molecule is being removed. Now, if you want to break down a polymer into monomers, you have a hydrolysis reaction. Hydro means water, lysis means breakdown. So you add water to your polymer and you break it down into smaller monomers or shorter polymer here. Okay, that was a general idea about the formation of the macromolecules. Let's start with carbohydrates. You already are familiar with carbohydrates, and we know we'll consume a lot of carbohydrates throughout the day. And they consist of mono means one, disaccharides and polysaccharides. Di means two, poly means many. And monosaccharides, obviously, we're talking about one Lego piece. They cannot be broken down. Important examples are glucose and fructose. Then we have disaccharides. Di means two, so obviously they're formed from 2 monosaccharides. Example would be lactose, which is the disaccharide that is found in our milk. Maltose is found in our beer, and sucrose, in fact, is our table sugar. Have you noticed that they all end with O's? So sugar usually ends with O's, anything that contains sugar. And then we have polysaccharides. So many of these monosaccharides are attached to each other to form a polysaccharide. Example would be cellulose. We don't have any cellulose in our body, or we can't digest it. Animals have enzymes that can digest cellulose. We do have enzymes that can break down starch. So starch is found usually in potato, pasta, rice. And there's also another polysaccharide, glycogen, that is usually stored in our muscles. So as you can see, a starch molecule consists of a large chain, and the monomers are glucose. So you have glucose, 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 and so on to make the starch. Same as glycogen, same as cellulose. So the glucose molecules get together through the dehydration process to form the long chain of starch. I'll let you guys to review this on your own. The next category would be lipids. So we covered carbohydrates, then we have the lipids. Lipids are hydrophobic. Hydro means water, phobic means they hate it. So as you can see in if you make when you make Italian salad, you can see that the vinegar separates itself from the olive oil because they don't like each other, so they don't mix. So this is pretty much one of the characteristics of the lipids. Now, lipids can be divided into fats and steroids. One of the major fats that you should be familiar with, and it's found in our diet, is the triglyceride. Each triglyceride molecule consists of a glycerol and 3 fatty acid chains. Okay, and they give us the energy that we need. And when you digest them, when you eat them, you break them down again into glycerol and fatty acids. And you also talked about on the discussion board about saturation and unsaturated versus unsaturated fat. Again, I'll let you discuss that because obviously any fat that is solid is not very healthy, and fat oils that are fluid, they're liquid, they are more healthy for our body. Now, I'm also passing through the other example for the lipids was steroids. Here you can see the basic structure of the steroids is the cholesterol, and you can see that estrogen and testosterone one of the differences is just the functional group. Like for example, you see that the functional group here is different than here, or the derivative that is attached to it, or the functional group here on the cholesterol and testosterone. So only a slight change in the cholesterol will cause estrogen or testosterone to be produced, but you can see a small change in the chemical structure causes major or a huge changes in the appearance. Now, steroids, you're familiar also with the synthetic anabolic steroids. Some athletes, even though it's illegal, they take them to make more muscles, to enhance their performance. Okay, we covered carbohydrates, we covered lipids. The next step would be proteins. Proteins, again, are also large molecules. They're made of amino acids. We have only 20 amino acids. However, when we put them together in different sequence, then we'll end up having millions of proteins in our body. So the definition of proteins is a long chain of amino acids, but we have only 20 amino acids, and one of the major proteins in our body is enzymes. So enzymes are chemicals, they're like catalysts in our body. They speed up chemical reactions in our system, and they're needed. And the enzymes usually end with ase. So the suffix for the enzyme is ase. So when you take sugar, table sugar, which is called sucrose, in order for your body to break it down to make energy from it, you need sucrase, the enzyme, to break down sucrose. Okay, going back to the amino acids, like I said, amino acids are 20 different types. However, the good thing about them is that they all have certain chemicals that are the same in the structure. So they all have a central carbon. This is the central carbon. It's just in the center. And there's a hydrogen atom attached to it. On the other side, there's an amino group. It's a functional group that is attached to it. On the other side, there's another functional group that is called carboxyl group attached to the central carbon. And then you see a side group. The side group could be only H, could be CH3, could be different kinds of chemicals that are attached to the central carbon. So what is different between these 20 amino acids is the side group. Like serine, one of the amino acids has CH2 and OH. Leucine has CH2, CH2. Do you have to memorize that? No. However, you need to know the common structure of all amino acids, meaning central carbon, hydrogen, amino, carboxyl, and a side group. So if you want to make a protein molecule, you have to do a dehydration from 2 amino acids. So this is one amino acid, another amino acid, and if you can see, the hydroxyl group from the carboxyl group is taken from the hydrogen group here, and the water, they make a water. However, once this molecule is being removed, you can connect the CO to the nitrogen, as you can see here. So these 2 amino acids are now connected, and the connection, remember that's a covalent bond, they're sharing electrons. It's called a peptide bond in amino acids. And remember, since we said proteins are a long chain of amino acids, this process can continue. So now another amino acid can bind to it to form a longer chain here. But again, you have to remember 2 amino acids through the dehydration reaction can form a dipeptide, 2 of these amino acids, and they're connected through the peptide bond. Another thing you have to remember that the OH is taken from the carboxyl group and the H is taken from the amino group. Okay, so proteins are very important in the structure and functions of our body. Obviously, structure function, if your hand is broken, it cannot accomplish the task. So the proteins also depend very much on their structure. So some proteins have a primary structure, meaning they're just a long chain of amino acids. Some have different other kinds of structures, like for example, this is a picture of a red blood cell, and red blood cells depend very much on their structure because usually oxygen gets attached to them, and that's the main function of the red blood cells, to carry oxygen within our circulation. Now, in sickle cell disease, patients who suffer from sickle cell disease, the structure changes, and therefore they cannot transport oxygen. So you can imagine if your body cannot transport oxygen, you're in trouble. And the reason for that is that one of the structure of the protein that is found within the red blood cell is being changed. And that causes the structure to be changed, and as a result, the function to be changed. Then, besides the primary structure, some proteins have the secondary structure, tertiary structure, and the quaternary structure. So, under the secondary structure, you have pleated structure, alpha structure. On the tertiary is the 3D structure, and the quaternary is when you have many proteins attached to each other. So again, the proteins are very much dependent. Their functions depends on their structure. Now, what causes the proteins to lose their function? Meaning, how do they lose their structure and therefore enhance their function? Excessive temperature. So very high temperature. If you boil proteins, and they can be denatured, meaning they lose their structure, and as a result, they will lose their function. Excessive pH. All proteins like to live in a certain pH environment. So if you change the pH of their environment drastically, again, they are denatured. We'll talk more also about that in the lab. So we cover carbohydrates, lipids, proteins, and the last category is the nucleic acids. Nucleic acids are also large polymers, and they're made of monomers. Remember that first piece of the Lego, and that is called the nucleotide. So a bunch of nucleotides attached to each other form the nucleic acids. Now let's look at the Lego piece itself, the monomer, the nucleotide. Each nucleotide consists of a phosphate group, a sugar group, and a nitrogenous base group. So an example for the nucleic acid is the DNA. DNA stands for deoxyribonucleic acid. So the Na comes from the nucleic acid. RNA is ribo.